Hello guys and welcome back to Excelsior Academy. So we were studying about the transport mechanism, right? So in the previous lecture, we talked about sodium channel transporters as well as about the potassium. Now in this lecture, we are going to discuss about the ligand gated channels. Okay. So these ligand gated ion channels are also known as ionotropic receptors. So again, they are group of transmembrane ion channel proteins which open to allow ions such as Na plus K plus calcium. Now you must be thinking, then what is the difference between this ligand gated and the yesterday what we have discussed about, right? So what you have to understand here is that if suppose a plasma membrane is there and your this ligand gated channels are there. So in this ligand, something else is coming and binding. And this is getting open for your Na plus K plus calcium 2 plus. Able to understand? Previously, it was the change in the membrane potential difference that was making the channel open. Here, it is the binding of the ligand, which is opening the channel for your ions like Na plus, K plus, calcium 2 plus, and Cl minus. Okay. Did you get the point actually over here?
Yes, ma'am.
Yeah. Okay. So it will pass through the membrane in response to the binding of the chemical messenger, which is your ligand, such as the neurotransmitter. For example, we can talk about the acetylcholine, which is the common neurotransmitter. Okay. Now, when a presynaptic neuron is excited and it releases neurotransmitter from vesicles into the synaptic cleft. So yesterday I was explaining, right? So if you have a presynaptic neuron over here, and then here you have got the postsynaptic, and in between you have got the synapse. So अब क्या होगा? From here in the vesicles, the neurotransmitters are released. These neurotransmitters will go and bind to these ligand-gated channels. Okay. For example, acetylcholine. This goes and binds to the acetylcholine receptors here. Okay. So the neurotransmitter then binds to the receptors, which is located on the postsynaptic neuron. Now, when these receptors are ligand-gated ion channels, they result in conformational change, which opens the ion channel. So eventually what is happening? You have got the ligand over here, to which the ligand binds, ligand channel रहता है, उसको ligand bind करेगा. Now, this binding of the ligand to this channel results in the conformational change, because of which what will happen? The channel gets open and the ions are able to pass through. Okay. Thus, in return, it results either in depolarization for an excitatory receptor response or hyperpolarization for an inhibitory response. तो जैसे कि कल हमने डिस्कस किया, depolarization मतलब क्या हो रहा है? आपका वो रेस्टिंग पोटेंशियल से लेस नेगेटिव होना. हाइपरपोलराइजेशन मतलब क्या? रेस्टिंग पोटेंशियल से और नेगेटिव होना. ठीक है. So yesterday we saw in that case minus 70 millivolt था आपका कल resting potential. Right now, if it becomes minus 55 millivolt, then it is the depolarization, यानी कि आपका signal pass हुआ. If it is hyperpolarization, then it is from minus 70 millivolt to your minus 90 millivolt. It means the response is inhibitory. वो response आगे नहीं गया. ठीक है? Clear? Okay. So now under the ligand gated channel, we have got two types. So nicotinic acetylcholine receptors, which is particularly responsive to the nicotine. The nicotinic acetylcholine receptor is also a Na plus, K plus, calcium 2 plus ion channels. Okay. अब देखो, whatever till now I was explaining from the previous lecture, it is given over here in this figure. Look at the figure now. Stop me guys in case you're not able to follow any point. Okay. So you got the nerve impulse. That nerve impulse resulted in the opening of your calcium channels. Okay, calcium channels open हुए, calcium from the outside it is going inside. Correct? Now this calcium resulted in the formation of your synaptic vesicles. These synaptic vesicles carry your neurotransmitters. Okay. Now in this case, the neurotransmitter is your acetylcholine. Clear? Now, with the help of the exocytosis, these vesicles diffuse to the plasma membrane of the presynaptic neuron. Okay, they get fused to it. So what is happening? The neurons, these neurons are released into the synaptic cleft. Okay, this is a synaptic cleft. Now what is happening? The neurotransmitters are going and binding to the ligand. So these are your ligand gated channel. तो जैसे ही यहाँ पे आपका ligand bind हो रहा है, so that results in the opening or the conformational change, which eventually results in the opening of the ligand gated channel, right? So what is happening then? Then those ions, for example, like Na plus, they will come inside of the postsynaptic neuron, and they are responsible for continuing the nerve impulse. Is this much clear, guys? This entire thing, because this is how in future also when we will talk about the Na plus K plus ATP channel, then also this is going to be a part of it. Okay. Now, Akshaya, any doubt over here? Would you try to explain this? Yes.
Yes, ma'am. So when the nerve impulse comes, the calcium channel is opened, and then the synaptic vesicle is formed, which carries the neurotransmitter. Then through exocytosis, the vesicles are going to bind the membrane, and then it is going to release the neurotransmitters. And these are going to bind to the ligand. When these bind ligand, the ligand channels open. The channels open, and then there is conformational change, and then it opens, and then the ions flow inside through the channel.
Yes. And then those ions are responsible for carrying the nerve impulse to the next neuron. Okay.
Huh.
Okay.
Okay.
Good. So these ligand gated channels are eventually at the end are your Na plus K plus Ca2 plus only. Okay. Yeah. Now the mechanism. So what all right now we have discussed in more detail, it is given here. Okay. So right now we are talking about the acetylcholine receptor. Okay. So here the mechanism, binding of acetylcholine to the N terminus of each of the two alpha subunits results in the 15-degree rotation of all the M2 helices. So M2 helices of what? Your ligand gated channel, right? Acetylcholine gated channel. I'll show the figure in the next slide. So what you have to remember is that the acetylcholine is binding to the N terminus, to the alpha subunits. Okay. So when acetylcholine binds, okay, there are two binding sites where acetylcholine comes and binds. So when it is binding, what is happening? It is rotating to 15 degree. Okay, so 15 degree rotate होगा, जिसकी वजह से क्या होगा? The closed channel over here will now become porous. Okay, hollow हो जाएगा, through which the ions can pass through. Okay, I'll show the figure. So the cytoplasm side of the receptor has rings of high negative charge, which determine the specific cation specificity of the receptor and remove the hydration shell often found by the ions in aqueous solution. So as we saw in the last time, तो अगर K plus है तो उसका भी एक hydration shell रहेगा, अगर Na plus है तो it will have its own hydration shell, right? Hydration shell. So depending on what ions it is responsible, the amino acids will be there present having the negative charge. Okay. Now in the intermediate region of the receptor within the pore lumen, valine and leucine residues, this is important, they define a hydrophobic region through which the dehydrated ion is going to pass. So in order to remove the hydration shell of the positive or the cations, that time you need negative amino acids, right? और फिर एंटर में sorry, then at the center where the only ions are present, okay, without any hydration shell, then the valine and leucine are helping them to pass through the channel. Okay, now look at the figure. So this is how it looks like your N-acetyl, sorry, not N-acetyl, the acetylcholine receptor. Okay, now look at over here. So it has got different subunits. Okay, so you can see alpha, two alpha subunits are there, then delta and gamma subunits are there. Now from the top you can see, so two alpha and one beta. Here the beta is there, okay. So on the top, if you see, you have got the two alpha, one beta and delta gamma is there. Okay, so these are the subunits of your acetylcholine receptor. Now, Look at this. So you have got two binding sites at the alpha subunit and terminals of the alpha subunit. Two binding sites are there of your acetylcholine, correct? Now, remember this negative charge also provided by the amino acids of these alpha helices, okay? Now, what you have to understand is that, let me just check whether I have got the another figure or not. Oh, it's not there. So what has happened is that here, the acetylcholine comes and binds, okay? Now remember, just now we have discussed. So the moment the ligand binds to its receptor, there is a 15-degree rotation. Now when this rotation is happening, the portion, this portion which was closed before, okay, it gets open up. Okay, and then the ions pass through. So I might have in the different figure. Okay, so this is the same which we just have discussed now. Okay, now you can see alpha subunits are there to which acetylcholine binds. The moment it gets conformational change, okay, the pore becomes it becomes hollow and Na plus is able to pass through. Clear? So this is in more detail of your acetylcholine receptor. So here they have also mentioned like how the neurotransmitters are being formed. Okay. So if you see, 1 second. Okay. So if you see we have got the choline is there, then acetyl-CoA is there. Then this is the enzyme acetylcholine acetyl transferase which converts into the acetylcholine neurotransmitter. Okay. ये भी थोड़ा सा पढ़ लोगे तो अच्छा है. So what I say is that this slide is more important than the others because in this everything and more information is given. Okay. So now what is happening is that these neurotransmitters are getting loaded into the vesicles, right? So if we want to inhibit this is the drug, Vesamicol. Okay, this is one step where you can stop the neuron transmissions. Okay, impulse transmission you can stop, or you can target these enzymes. ये एक हो गया, ये दूसरा हो गया. ठीक है? Then we have got the another one, that is fusion of these vesicles to the plasma membrane. So you have got the another toxin, botulinum, which is going to prevent your exocytosis. Clear? Okay. Now, if suppose it is getting released, now this is present in the synaptic cleft. Okay. Now, what happens? It goes in binds to its receptor, it gets opened up, and only Na plus K channels gets excited. Okay. Now, what happens to the remaining neurotransmitter which is present in the synaptic cleft? जो बाइंड नहीं हुए हैं तो उनको क्या करना होगा, हमें उसको रिमूव करना है फ्रॉम द सिस्टम. सो देन यू हैव गॉट द acetylcholine esterase. Okay, so this is going to chop off your acetylcholine into choline plus acetate, and then these will get recycled. अब अगर मुझे यहाँ inhibit करना है, then I have got the ACH inhibitors. Clear? Now, this is one type of receptor which comes under the ligand gated. The another one is muscarinic ACH receptor. So both nicotinic and muscarinic, okay, इन दोनों का ligand आपका acetylcholine ही है. Okay, but the only difference comes in about like how and what kind of channels are getting open up. So if it goes and binds to the nicotinic receptor, then it is excitatory. ठीक है? अगर muscarinic में जाके bind करेगा and it activates your phospholipase C का activation, then it is excitatory. But if it goes and activates adenylate cyclase, जहाँ पे the enzyme adenylate cyclase is going to increase the concentration of cyclic AMP from ATP. If you remember in the previous lecture we had talked about, then it is going to be inhibitory. Okay? It will take time, 2-3 times you have to make the diagram to understand. So overall what you can remember is these drugs which are going to inhibit this nerve impulse and how the neuron transmission is happening. Okay, so far so good guys. Shruti, Tanvi, you guys थोड़ा सा late join किया, but I hope you are able to understand. ये पूरा समझ में आया?
Yeah.
Yes, Shruti?
Yes, ma'am, समझा.
Okay, your voice is very low. What about you, Tanvi?
No, ma'am, not that much clear.
Okay, do you want me to repeat once again?
Yeah, ma'am.
Okay, okay, no problem. Okay, so till now we were discussing about the ligand gated channel, right? So here the example is of acetylcholine ligand gated channel. Okay, so इसमें two types are there. So one is a nicotinic acetylcholine receptor, the another one is muscarinic acetylcholine receptor. So it just depends on the type of channels which is getting open, which decides that whether कि न्यूरॉन आपका एक्साइटेटरी होना चाहिए. एक्साइटेटरी मतलब इट इज गोइंग टू पास टू द अनदर न्यूरॉन्स. ठीक है, और अगर इनहिबिटरी हुआ तो इट इज गोइंग टू स्टॉप ओवर देयर. तो जो भी काम जैसे नहीं करना है तो आपको ब्रेन सिग्नल देगा, राइट, कि ये काम वहीं रुक जाना चाहिए. सो रुकना मतलब नो मोर सिग्नल शुड बी गिवन, राइट. So that depends on the channel which gets open up. Now here you have got the acetylcholine as your neurotransmitter. So this neurotransmitter is being synthesized by the enzyme choline acetyltransferase from the acetyl-CoA plus choline. Now these acetylcholine neurotransmitter, they get loaded onto the vesicles. Okay. Now these vesicles then get fused with the plasma membrane in the process called exocytosis. ये भी हम आगे जाके पढ़ेंगे, but I'm sure you guys know this thing. So exocytosis से वो fuse हो जाएगा plasma membrane में. Then from the from the presynaptic neuron, it comes to the synaptic cleft. Correct? Now from the synaptic cleft, now it will go and bind to its receptor. So once it goes to binds and its receptor, nicotinic acetylcholine receptor, हमने अभी देखा कि वो अल्फा सब यूनिट में दो जगह पे बाइंड होता है. ठीक है, और जैसे ही बाइंड होगा 15 डिग्री रोटेट करेगा, जिसकी वजह से वो जो आपका रिसेप्टर है ओपन हो जाएगा एंड इट विल ओपन योर Na+K+ चैनल. ठीक है, तो आपका जो भी Na+K+ जो भी चाहिए वो आ जाएगा एंड सिग्नल विल बी पास. If it goes and binds muscarinic receptor, then अगर adenyl cyclase activate होता है, then it is going to increase the concentration of CAMP. CAMP बढ़ेगा तो आपका वो inhibitory होगा. Okay, this is another mechanism binding to the another receptor. Now, what else is required is to remember. एक हो गया आपका hemicholinum, which is going to block the uptake of choline through this channel. Another one is vesamicol, which is going to inhibit the uptake of the neurotransmitter into the vesicles. Okay. Botulinum toxin, which is going to inhibit the exocytosis of this vesicle to the plasma membrane. Then we have got the acetylcholine esterase inhibitor. So इससे क्या होगा? The acetylcholine will remain in the synaptic cleft. Okay. जिसके वजह से वो continuously excitatory stimulation देगा. Okay. So if I don't want, then I need this enzyme to degrade the excess of the neurotransmitter. Clear, guys? Now, थोड़ा बहुत clear हुआ? So 2-3 times when you go through this figure, you will be able to understand more. Okay. Can we now little bit clear it is?
Yeah, ma'am.
Okay. Okay, now this is about the second, which I just mentioned now about the muscarinic acetylcholine receptors. So पहले कैसे था, वो निकोटिन को रिस्पॉन्स करता है, यहाँ पे वो मस्करानिन को रिस्पॉन्स करेगा. इट इज जस्ट नथिंग बट द सिंपल ऑर्गेनिक कंपाउंड ओनली. सो अगर स्टिमुलेटरी है, देन इट इज गोइंग टू एक्टिवेट योर GPCR. ठीक है, ये भी हम पढ़ेंगे इन डिटेल. जी प्रोटीन कपल्ड रिसेप्टर्स, इट इज गोइंग टू एक्टिवेट योर फॉस्फोलाइपेज C. फॉस्फोलाइपेज C क्या करेगा, आपका जो IP3 है, दैट इज योर 1,4,5 आइनोसिटॉल जो है आपका, वो उसको डिग्रेड करेगा एंड कैल्शियम रिलीज होगा. ठीक है, दिस इज वन मैकेनिज्म जहां पे वो स्टिमुलेटरी है. नाउ कमिंग टू द इनहिबिटरी. इफ इट इज इनहिबिटरी, देन the GPCR which gets excited is I for the inhibitory, which is going to activate adenylate cyclase enzyme. Okay, and then the concentration of CMP is going to describe like whether it is inhibitory or stimulatory. Okay, now yeah, so it is not the ion channels but belong to the superfamily of GPCR which activate the other ionic channels via secondary messenger cascade. तो इसका क्या मतलब हुआ? कि ion channels directly open नहीं होते हैं, but it is the action of the GPCR which is helping the other ionic channels to get open up. ठीक है? So the muscarinic cholinergic receptor activates G protein when bound to extracellular ACH. So the alpha subunit of G protein activates guanyl cyclase, while the beta gamma subunit activates K channels, therefore hyperpolarize the cell. ये आपको और क्लियर हो जाएगा when we will talk about the GPCR in the cell signaling. ठीक है? कैसा है ना, everything is connected, so you just can't talk about one particular receptor at one time, so you need to include the other part also. Anyways, so we'll talk about GPCR in the next lecture more. बस इतना याद रखो, अगर alpha subunit of GPCR है, so it has got again 3 subunits are there. ठीक है? GPCR में alpha, beta, gamma है. alpha एक साथ है और beta gamma एक साथ है. If alpha gets activated, then it is stimulatory. बीटा गामा अगर एक्टिवेट होते देन इट इज इनहिबिटरी. ठीक है, इतना आप याद रखें. ओके, सो दिस इज आल्सो द रिस्पांसिबल फॉर द डिक्रीज इन द कार्डियक एक्टिविटी. Now let's talk about the mechanically gated channels. Okay, so mechanically gated channel, this is I think only the one example I have here. Okay, so it is a class of ion channels which is capable of responding to mechanical stimulus. For example, mechanosensitive ion channels are there, which gets open under the influence of stretch, pressure, shear, and displacement. Okay? For example, here we have taken the example of internal ear of a human. Okay? Abhi, I don't know how are you going to visualize this thing, but if this is a structure of the internal ear, you might have seen that there are certain small hairs present on that, and each hair is connected to the auditory nerve fibers. Okay, now the tympanic membrane हम नहीं बोलते जहाँ पे आपका sound गिरता है, it vibrates and that's how you're able to understand the signal. थोड़ा बहुत मालूम है क्या, like how are we able to hear the stuff, right? थोड़ा सा idea है that how are we able to listen. So when the sound waves fall onto the tympanic membrane, okay, it vibrates. Now that vibrations are nothing but connected to the nerve fibers. Those nerve fibers are giving the signal to the brain. Okay, this is the sound which is now being put on your onto your ear, and इसका ये मतलब है. ठीक है, अभी जैसे जैसे मैं कुछ बोल रही हूँ, okay, suppose I say ear, so the sound which is falling onto your internal eardrum and all, it is vibrating in such a way that your brain recognizes that right now what you have heard is the word ear. Okay, now how these receptors are present, so you can see these are the supporting cells are there, and each auditory hair cells what you see, okay, they all are connected to the auditory nerve fibers. So when there is a sound waves coming, oh, 1 second. So when there is a sound waves coming, what is happening then? ये देखो यहाँ पे close है. So when the sound is coming, what is happening? They're getting tilted. Okay, because of the waves, it is getting tilted. उसके ऊपर pressure आया ना एक तरह से, तो आपके ये जो channels हैं, they get tilted. Now when they get tilted, because of this, the channel gets open up. So when this channel gets open up, the ions will go inside, and it is going to give the signal to your brain and to the nerve fibers what kind of sound has been falling onto the internal ear of the human ear. Okay, thoda sa idea aaya. What I can suggest over here is that as a Java information is not available based on the mechanical gated channel, if possible try to watch certain videos, like you can go to YouTube and watch. थोड़ा सा आइडिया मिलेगा कि कैसे आपका कान कैसे, like how are you able to hear it, okay, and how these mechanical pressure or the stretch which falls on the eardrum is actually opening these mechanically gated channels. Okay, I hope this you're getting little bit idea over here. Anyways, question तो कभी नहीं पूछा mechanically gated channel में से, but it's just for your information. Yeah, anything else guys? Is it clear?
Yes, ma'am.
Yes, ma'am.
Okay.